Shortly after the turn of the century, and just after viruses and cells were discovered, the French scientist Borel suggested that these viruses might have something to do with cancer. But his suggestion was not taken very seriously at that time, and nothing was done about it. Then about 5 years later, Ellerman and Bang, working with a fowl or a chicken leukemia, found that this disease was caused by a virus. However, because this disease was not then generally regarded as a malignant disease, this suggestion had no influence on the virus-cancer relationships. It remained for Peyton Rouse, working with chickens, in 1911 to discover the first virus-cancer relationship. Dr. Rouse found that a chicken disease, which you can see here, a large growth on a certain chicken, was caused by a virus. He took a growth such as you see here, removed it from the chicken, and used those cells first to inoculate other chickens, and found he could cause this disease. And then he made the really surprising discovery that he could make extracts of this tumor, pass it through a filter which would hold back all living organisms, and he could then cause the cancer in chickens. Now, this is a natural disease of chickens. It's not a man-made or laboratory-made disease of chickens. Dr. Rubin, working our laboratory, has been able to use this same virus and show that he can measure the amount of this particular virus by growing it not on whole chickens, but on the cells of chick embryos, such as you see here. Here is a layer of chick cells in this little dish. And if we could look at it closely, you would see that there are little cancers in this layer of cells. In order to look at it closely, we've taken, and here is what you see if you look at it under the microscope. You see these two large spots of cancer cells. You have literally, in a dish like this, these microcancers caused by the Rouse sarcoma virus. Let us now take a look at a film in which we show first some normal cells, actual cells, and then an animation, and then some cancer cells, and see how these cells multiply. Here you see the normal cells of a tissue going all the way down to the basal layer where these cells are formed, and you see the cells dividing. Here is an actual cell in division. You see the chromosome separating, a new set being produced, and the cell then dividing. This is an actual picture of a cell. And here you see this cell multiplying and going up through and sloughing off cells at the exterior, in this case of the skin. There's a very rapid multiplication of these normal cells. Now here is a picture of a cancer cell in which you see the cell dividing gradually, but instead of dividing normally, it produces many, many units. And this cell grows wildly, as you can see, and produces a cancerous growth. This growth extends all the way up through and makes a large tumor. Here again is a cancer cell that you can see dividing. Instead of dividing into normal 2 cells, you're getting 3 or more individual cells. This is an actual picture of a cancer cell dividing. And here again, you can see that the growth is wild. Here it is in animation, and the growth just springs up through and forms a large cancerous growth in the normal tissue. One of the things which has interested scientists is the fact that they have found that there is great variation in the actual amount of virus that's present in these virus-induced cancers. Now, another virus that's been interesting to us is the Shope rabbit papillomavirus. This virus has been obtained in quite high purity, as you can see here from these electron micrographs, which show a variety of viruses. And the one we're interested in is this one here. These are purified Shope rabbit papilloma viral particles. If you look at these close up, you see here they are. One of these particles is sufficient to cause a cancer in rabbits. Another example of the high specificity of viruses is exhibited by a virus that Dr. Luque worked with. It's known as the kidney carcinoma virus of the leopard frog. Dr. Luque found that when he inoculated this virus into a frog, unlike what you might expect, that is to get a cancer at the point of inoculation, the virus always caused a cancer in the kidney. Actually, you can put the virus into the skin, into muscle, or here, there, and yonder. But wherever you put it in, it always goes directly to the kidney and causes a cancer there. In other words, here is an example of high specificity in that the virus goes only to the kidney cells of the leopard frog. It will not cause disease in certain other kinds of frogs or in other kinds of animals. Another virus of this general type that causes cancer was discovered by John Bittner. It has come to be known as the mouse mammary cancer virus. This virus is very interesting because it is thought to be passed in the milk from the mother to the offspring. And when the mouse gets a little old, you can find that it will come down with great tumors formed around the breast. Here is one which is almost a third as large as the mouse itself. It's interesting that the incidence of disease in mice by this particular virus can be varied over very large ranges. If you change the genetic background of the mouse, in other words, you breed certain strains of mice, and if you change the amount of hormonal stimulation, the incidence or the amount of cancer that you get can be varied from almost 100% down to about 10%. So this presents some interesting opportunities for research. Since Peyton Rouse first discovered the sarcoma virus of chickens, a great many cancers of animals have been found to be due to viruses. These cancers occur not only in chickens, but in ducks, in mice, in frogs, in rabbits, cats, dogs, horses, cows, deer, and in still other animals. It's interesting to note that there are some plant growths that are caused by strains of tobacco mosaic that are quite irregular and have many of the attributes of a cancer. Well, despite all of this vast amount of evidence which relates cancers in animals to viruses, there's been a great reluctance to accept the possibility that human cancer might be due to a virus. Now, this is due perhaps largely to the fact that we have no evidence that human cancer is infectious. However, I believe that basic biological properties do not generally differ very much as you go from the cells of one species to another. In 1956, when I had an opportunity to speak to several hundred medical doctors, I expressed the opinion that the time had arrived when we should simply assume that most cancers are due to viruses and plan and execute our experiments accordingly. During the past several years, scientists have made great advances in techniques for concentrating viruses, so that if the fact that nobody has yet obtained a human cancer virus might be due to the low concentrations of virus in such cancers, as obtains occasionally in the case of the Shope rabbit papilloma cancer and in the case of the Rouse sarcoma. Then these new concentration techniques should now be applied to possible human cancer viruses. However, there is still a roadblock, and this has to do with specificity, with the fact that most viruses will cause disease only in certain kinds of cells. And obviously, we cannot make tests of possible human cancer viruses by deliberately inoculating other human beings. Well, in the last few years, this roadblock has been eliminated because of the cultivation of a whole variety of cells in the laboratory, in the test tube, so to speak. Elsa Zitzer, in our laboratory, found that the human amnion, this is the membrane that surrounds the human baby at birth, can be grown in tissue culture. Here is such a human amnion, and you can take this membrane, which consists of living human cells, treat it chemically to break up the membrane a bit, and then grow these cells in the laboratory. Here, for example, is a dish in which some of the cells of the human amnion are growing. Now, the layer of human cells in this dish can be used to grow viruses, and a variety of viruses have now been grown in human amnion cells. From the standpoint of cancer research, it's been interesting that as you continue to grow the human amnion cells in the laboratory, after a few passages, something happens to them, and they seem to become more like cancer cells. Well, obviously, we are studying this transformation from normal human cells into cells that have certain properties of malignancy. Well, you may wonder if there is this possibility of viruses being responsible for human cancer. Why hasn't somebody found a cancer virus, a human cancer virus? Well, actually, in the past 5 years, 200 new viruses causing disease in human beings have been found. And in some cases, it's not yet been possible to associate some of these viruses with a given disease in the human beings. And one wonders whether or not they may or may not have anything to do with human cancer. Well, in view of the fact that human cancer does not appear to be infectious, it may be necessary for us to think along certain other lines, to seek analogies that may give us an insight into possible new relationships between viruses and human cancer. And for this, we might think just a moment about what takes place in the bacterial virus field. Here we know that a bacterial virus can infect a cell, and sometimes it can change itself a little bit, take on a new property, and instead of bursting and killing the cell, it lives with the cell. And each time the cell divides, the virus divides. This is a phenomenon we call lysogeny. Well, now, such lysogenic bacterial cells, when subjected to certain shocks, such as X-rays or certain chemicals, the virus can be activated, and then it springs into action, multiplies many hundreds of times over, and causes disease. In this connection, it's interesting to note a disease of mice, mouse leukemia. You can take some mice and examine them very carefully for a mouse leukemia virus, and you do not find the virus. And Ludwig Gross and also Henry Kaplan found that through the treatment of such mice with a few amounts of X-rays, that the mice would then come down with leukemia. And then, most surprising, when they examined the tissues of such mice, they found that their virus was present. They had brought out a hidden leukemia virus, brought it out into the open, and now this virus could be used and transmitted from mouse to mouse. Another possibility that we like to think of to explain the possible transmission of an agent that might cause human cancer is the business of what we call transduction. Now, in transduction, a virus can go from one cell to another cell and apparently transfer a bit of genetic material from the cell that it first infects to a cell that it infects later on. Let me show you, for example, what would happen if you infect a bacteria, which is shown here, by means of a bacterial virus. You can have the viral particle come up, and as you know then, some of the nucleic acid comes out and gets in there, and then it starts multiplying many, many times over with a lot of nucleic acid being produced, and this multiplication is eventually followed by the production of a large number of bacterial viral heads, and then finally the complete virus. Well, now it's known that during this multiplication in transduction, one of the nucleic acid particles seemingly has the ability to take on a little bit of genetic material from this cell. And then when this cell bursts and the particles are spread all over, this material, which is now present in one of these particles, can come and infect still another cell. And when this takes place, it carries into this cell this bit of genetic material. And then when this cell multiplies, it carries a genetic trait which is characteristic of this particular bit of material which is introduced into this cell. And then when the cell multiplies, as you can see here, it produces cells of this type. This type of cell is a type of cell which is characteristic of the cell from which the virus was first obtained. So that in this process, we see that genetic material is introduced from one cell into another cell. Now, if by chance this little bit of genetic material should have a cancer tendency, it would be possible for cancer thus to be transferred from one person to another. But since this takes place only once in about 100,000 times, you can see that it would not appear to be an infectious process. Well, during the last few years, there has been a great increase in the interest in the possible relationships between viruses and human cancer. Many laboratories are experimenting and attempting to isolate such possible human cancers. And in some cases, some rather interesting results are being found. For example, Dr. Leon Demachowski has made electron micrographs of sections, in one case, of a mouse suffering from mouse leukemia, and he finds particles such as those that you see here. He has also made electron micrographs of sections from the lymph nodes, in this case of a little girl who died of leukemia, and here he found these particles, very similar to those which occur in mouse leukemia. Well, we do not know whether or not these particles have anything to do with the cancer. This represents one of the new problems for the future. It is necessary first to prove that such particles are viruses, and then secondarily to prove that they are important or that they are the agent that causes the cancer. Well, during the past few years, scientists have made vaccines against some of these animal cancers, and this means that this offers a possibility for the future in case we find that there are viruses that are responsible for human cancer. Now, one of the important areas of research has to do with the chemotherapy, and much of this has to do with the work on building blocks similar to those of nucleic acid. Because the heart of a virus is the nucleic acid, this brings us together in virus research and in chemotherapy. Well, all in all, it's well to remember that so far as we scientists are concerned, there is only one fast, direct way to produce a cancer, and that is by means of a virus. We're really only at the beginning stages of this, and much more needs to be done in the future. And I believe that this represents an area of rich opportunity for experimentation. And there are many, very interesting problems for us there. Well, now I'd like to summarize just a moment about viruses generally. As you know, some of these viruses can be obtained in crystalline form. And we have here a crystal of such a virus which has been cut through by means of a knife and then looked at by means of the electron microscope. Each one of these little particles that you see here represents poliomyelitis virus. And this is of the magnification of about 1,000,000 times. And if you enlarge one of those particles still again and make a model of it, representing much of what we know, this is a model of a single particle of poliomyelitis virus that causes disease in animals and in human beings. And over here we have a model of a plant virus, tobacco mosaic virus, showing the protein subunits and the very interesting structure that it has. And then here on the floor we have a drawing of the third great class of viruses, the bacterial viruses. And now I'd like to have my colleague, Robley Williams, tell you something about the implications of virus research for the future.
It's well to recall the primary differences between viruses and other organisms that produce disease, such as bacteria, because these differences really represent the heart of the interest that virologists have in viruses. They're two in character. Viruses are much simpler chemically than bacteria, consisting only of nucleic acids and proteins. And secondly, and more importantly, viruses can grow only inside living cells, whereas bacteria can be cultivated in the test tube. Now, when there is a virus infection, what goes on? Well, a virus particle or part of a virus particle invades the cell. It gets inside. Then after a certain length of time, one finds that a great number of virus particles of entirely similar nature come out of the cell. This is the process that we can observe, and what goes on in between the going in and the coming out is largely the mystery. Now, the cell may react in different ways. In some cases, the results are quite dramatic. The cell is healthy at the start, but is entirely dead and badly damaged by the time the viruses get out of the cell. While in other cases, such as the tumor viruses, the cells are actually encouraged a little bit to grow even better, although at the same time they are, to be sure, producing viruses. But now, what goes on? What sort of questions can one ask concerning this mystery time between the time of infection and the time of virus release or cell death? I can ask a lot of questions. For example, does the nucleic acid that goes in come apart? Does it come into 8 or 10 or 12 short pieces of the original nucleic acid, or does it stay together? Where does the nucleic acid go when it comes into the cell? Does it stay just in the outside of the cell? Does it go in toward the nucleus? If so, what propels it? Does it just rattle around with a sort of Brownian motion, or is it pulled somewhere by a force? Is it necessary for the nucleic acid of the virus actually to find the nucleic acid of the cell and somehow or other make close contact in order to so change the nucleic acid of the cell that the cell now becomes essentially a virus factory, making viruses instead of its normal product? Now, these are only rather specific questions which in a more general way can be asked about the cell itself. How is it that in a cell, the nucleic acid found in the nucleus of the cell, mostly in the chromosomes, is able to regulate the mechanism of the cell? Why is it that we have heredity? Why is it that one cell, which is producing 2 daughter cells and then 4 daughter cells and so forth, why is it that these cells retain their essential chemical and biological characteristics? We don't know any of these answers. Our area of ignorance is obviously far greater than our areas of knowledge. But we virologists believe that through the study of viruses, we may be able to answer, at least in a peripheral way, some of the very complex problems of life. Because the viruses are relatively simple. They are the only object from which we can remove the life stuff, namely the nucleic acid, and purify it, and chemically modify it, and then put it back in the cell and have it biologically active in the sense that it performs just as whole viruses do. So that we have here essentially a biological probe, and that we can hope to determine how the structure of the nucleic acid affects what we would call the heredity of the viruses. Now, we have reason to believe that it is the sequence of the building blocks of the nucleic acid that determines the sequence of the building blocks of the proteins, and that the manufacture of the proteins is determined by the structure of the nucleic acid. So if, in the case of viruses, where we can purify the nucleic acids and the proteins, if we can determine in great detail the sequence of the building blocks, both for the nucleic acids and for the proteins, we should be able to find essentially the answer to a coded message, namely, how does the one sequence affect the sequence of the other? Now, what is about to happen in the future with respect to virus structure and the biological implications of virus structure? And I think what will happen first is that in the long run, we will succeed in determining the detailed structure of nucleic acids. We will succeed in determining the detailed structure of proteins. We will see how the two are related. With anything like luck, we shall eventually be able to make viruses, or at least make the nucleic acids of the viruses. Because after we have found out how they are constructed, we should eventually be able to understand how to put together the building blocks. And I'm willing to bet that if one puts together the nucleic acid of a virus in the way it ought to be put together, then if one tries to infect a cell with this nucleic acid, he will indeed have made the nucleic acid of a virus, and so will have made what one might call the simplest form of life. So through viruses, we may be able to unscramble quite a few of the mysteries of living cells. And so for us as living beings, this is essentially the challenge of life.
Virus was produced for the National Educational Television and Radio Center by KQED San Francisco, in cooperation with the Virus Laboratory of the University of California at Berkeley. Featured in this program were Dr. Wendell M. Stanley, Professor of Virology and Director of the Virus Laboratory, and Dr. Robley C. Williams, Professor of Virology and Associate Director of the Laboratory. The series was made possible by a grant from the National Science Foundation. This is National Educational Television.